I'm really pleased to be able to turn things over to our first workshop of eCOTS 2020, entitled Multivariable Thinking Across the Curriculum. And without further ado, I wanted to introduce our speakers and presenters today. Jamie Bunting and Elena Keeling are from the Department of Biological Sciences at Cal Poly San Luis Obispo. Karen McGaughey is professor in the Department of Statistics, along with Beth Chance. And Nathan Tintle is a professor and director of the Keeling Center for Research and Scholarship and a professor of statistics at Dordt University. I think I got everybody on this stellar team. This group has been doing lots of things in so many different ways and places. Two things in particular I'll just highlight right now is the STUB grant. This is the statistical thinking in undergraduate biology and biological sciences. And the goal of that grant is to bring together the biology and statistics faculty to collaborate on statistical education of biology students and to ensure that biology contexts are incorporated for stat students. They've also been actively involved in the intermediate statistical investigations, a forthcoming second course text that focuses on explanation of variability and multivariate thinking. So without further ado, I'm going to turn things over to Nathan Tintle and this, again, great esteemed group of folks. Just a reminder, questions can go into the Q&A, other comments can go into the chat window. Take it away, Nathan, it's all yours.
Thanks, Nick. Thanks for the great introduction. Good morning to everyone. I think it's morning, at least in a lot of the time zones here today. I'll probably stop my video here in just a minute to make sure that we've got a good internet connection and you can see my slides. So I think what we'll do is I'll give a brief introduction, some motivation, and then we'll take a minute here and I'll let each of the other presenters introduce themselves a little bit more as well. But really, you know, our motivation is that for far too long, early courses in statistics education have focused on one or two variables at a time. The newest GAISE guidelines for introductory statistics do talk about a multivariable thinking experience in the first course. But really, how should we do this? This is a bit new, and people are doing different things. And really, what does this look like in the first course, but then beyond the first course as we look at a second course and even beyond that? It's this thinking that we're trying to sort of explore here in this session today. And so before we get too much further, what is multivariable thinking? And these are some ideas that our team has come up with as we think about this term. Recognizing that variation in a response variable comes from many, many sources and being able to identify those sources. Recognizing that relationships which are discovered in the statistical analysis or data visualization depend in large part on how the studies dealt with many other variables that may influence that response. Designing studies that effectively control for many variables, things like blocking and inclusion criteria. Analyzing data in the presence of confounding and bias, and then drawing appropriate conclusions from data analysis and conveying the limitations of these conclusions. So these are some of the things we think of when we think about what is multivariable thinking. And so our goals today are to appreciate why multivariable thinking is so important for all statistics and non-statistics students across levels. Hopefully to get you thinking about how you can approach multivariable thinking in your courses, and to get you thinking about what we're going to call vertically integrated approaches to multivariable thinking within and beyond your department. So first course, second course, and beyond. How does sort of the ways you talk about multivariable thinking vertically integrate across those different courses? And so our approach in this session to sort of get you towards those goals is to talk about some additional motivation for a focus on multivariable thinking versus other topics. We'll talk a little bit about some curriculum and pedagogy projects we're working on that are being impacted by a focus on multivariable thinking and give you some concrete examples you could think about for your own courses. And then to have plenty of time for questions and answers with all of you to hear what you're doing, to ask us questions, to brainstorm, think, and dream with you about the future of multivariable thinking in statistics education. And so even though we're in this online setting, we do hope that by having good, healthy Q&A times, we can sort of replicate what it would be like if we were in person and have some good conversations together. So here's the session overview. We're in this introduction time right now, trying to do a little bit of motivation, goal setting, introduction to presenters. We'll do that here in a minute. I'll then take a few minutes and do a little bit of a deeper dive into motivation, some problems we're seeing in practice, why multivariable thinking versus other topics. And then we'll get into the meat of the workshop. And we've broken the workshop into three main pieces. We're going to dig deeper into three main parts. The first one is, how does multivariable looking or how does multivariable thinking look in discipline-specific courses? So we're going to dig into a case study in biology, some things that are going on at Cal Poly. Then we'll go to a second part on a second course in statistics. And then the third part will look at a first course in statistics. So we're going to kind of start with the end. So what do we want to do in those discipline-specific contexts with students who are heading off after their formal statistics education? And then we're going to walk back in the statistics curriculum to hopefully show you this kind of vertical integration as we go, kind of starting with the end in mind. Where do we want students to get to when they're done with their formal statistics education? And then how can we lay the foundations and lay the groundwork for that in the earlier courses? And then we'll take some time at the end for some wrap up, pointing you to some resources, acknowledgements, and final Q&A. You'll notice that we have Q&A built in after each of those main three parts of the session. And so we'll do some Q&A as we go. I do want to take a minute now and let each of our presenters briefly introduce themselves, and we'll just go in the order that's on the screen there. And so Beth, do you want to just give a brief introduction?
Hi, I'm Beth Chance. I'm a professor in the Department of Statistics in surprisingly rainy San Luis Obispo at Cal Poly.
I'm Jamie Bunting. I coordinate the biology laboratories, the intro biology laboratories at Cal Poly.
I'm Elena Keeling. I'm a professor in biology at Cal Poly.
And I'm Karen McGaughey. I'm a professor in statistics at Cal Poly.
And hi again, I'm Nathan Tintle, and I'm a professor of statistics at Dordt University in Iowa, and also do a fair bit of work analyzing biological data on a number of different projects. So in terms of logistics for the session, as Nick already mentioned, but just as a reminder, please use the Q&A feature to enter questions throughout the session. Beth and all the presenters will help to moderate things so we can do some Q&A with you as we're going. So if you got questions or comments, the other panelists can jump in and interject with whoever's speaking if there's a question we want to sort of address along the way. But we also have those 4 set-aside times for Q&A spread throughout the session. So please ask your questions anytime. And also just feel free to add comments, sharing your approaches and ideas. You can also use the chat feature to ask technical questions. So if you're having any technical concerns or anything like that, Nick Horton and others will help monitor those. So now let me just dig in for a little bit more during this intro time on motivation for our session. And so a few years ago, I was talking with a biologist, and we were talking a little bit about some of the curricular things we were trying in the classroom. And she told a little story. She said, when I was an undergraduate, I actually kind of enjoyed statistics, but I left the course thinking that statistics was really useful for analyzing the colors of M&Ms and weighing M&Ms and counting M&Ms. And that it wasn't until I got into finishing my PhD and into practice as a biologist that I really understood how incredibly powerful and important statistics is. And so that there was this disconnection from reality, and it really challenged me in my thinking about teaching to make sure that we were moving, and the GAISE guidelines have been pointing us in this direction for a long time, moving from just hypothetical data to real data, but then even further to real data that matters. And so M&Ms are real and they're fun. And even in my own class, we still do some of these, kind of gimmicky things once in a while with students. But I would challenge us all to think about, do M&Ms, sort of really matter? Is this really the best way to illustrate to our students what's going on with statistics? And as we get into these more real data that matters contexts, we need to realize that the world is really complex. And the way we would quantify that or summarize that as statisticians is we would say that the world is very multivariable. And so we do our students a disservice if we gloss over this complexity too often, if we too often compartmentalize their learning and bring them into incredibly hypothetical, controlled scenarios. But on the flip side, we also do our students a disservice if we throw them into the deep end too quickly and expect them to sink or swim. And so we're doing this dance here of complexity. And one way to sort of visualize this, there's a lot going on the slide. This is a figure from a paper we published a few years ago, and I've added a few additional annotations here. But the argument we made in this paper and what we're trying to demonstrate in this figure is that it's really hard to get students towards statistical thinking. Where students want to go are one of two places. They either want to go into what we call the bog of overconfidence, thinking that statistics is like math, providing us with indisputable facts about the world we live in. Things are black and white. I will analyze my data. If the p-value is small, I will know what to do next. If the p-value is big, I will know what to do next. There's a right or wrong answer to the questions we're investigating. On the other hand, this other bog is the bog of disbelief, and that's that statistics, like magic, can be made to say anything. And so, practically speaking, it's very irrelevant. So I think that for the first bog, the bog of overconfidence, that when we give students oversimplified examples that are separated from reality, they're too hypothetical, it's too much M&Ms, that we might lead students to believe that statistics is black and white, right? And we can think of other ways this might happen. Maybe it's by emphasizing a p-value criteria too much in our classroom. Right? Too much push the buttons on the calculator and get a right or wrong answer. On the other hand, in this other bog of disbelief, if we give our students too many complicated examples, where there's lots of black box wizardry, click buttons on the computer, hope for a miracle, not really understanding what's going on behind the scenes, I think that we can lead our students to think that statistics is magical. It can be made to say anything you want, and so it's irrelevant. And so it's really hard because our students want to fall into one camp or the other. And I think society at large, people want to fall into one camp or the other. And so we want to try to push students, and they're represented with this circle, sort of this ball, up the top of this hill to the top of this hill, which is really the pinnacle of statistical thinking. That's keeping these two bogs sort of in check, where statistics is informing a rigorously scientific approach to problem solving and decision making. George Cobb, as you know, passed away recently, and he's been a core member of our team for a long time. And so I did want to, and many of his accolades are on the screen, I did want to use an analogy that George developed and that we've shared at other times, but just to sort of remember George and his thinking on some of this. And he said, you know, few students ever leave our course seeing statistics as this beautiful way to make sense of the world we live in. And so if we think of that pinnacle of statistical thinking, few of our students are leaving our courses up at the top, understanding these complexities and the beauty that statistics can provide in terms of making sense of the world. Maybe some of the better students get a fuzzy impression. But so many students are so close to the canvas that all they see are disconnected details. And I think when our students are left in one of those bogs, they're seeing disconnected details. Things are so complicated, they don't make sense, and they don't know how to proceed. And so we think that in order to keep students focused on statistical thinking, we need to emphasize multivariable thinking. So what does this mean? It means a focus on explained variation. It means a focus on the logic of inference, the overarching process of statistical investigations. It means a focus on what statistics can and can't tell you. And it's a focus on practical problems. Not all the time is spent on data cleaning and data import. They're practical, and they're walking this fine line of not oversimplified, but not too complicated. And we know that those things are sort of held in tension. So let me say a couple more things. Part of our motivation is also from what we're seeing in practice. And I consult on a lot of different projects where I'm a collaborator or a consultant. And these are a couple anecdotes that have come up for me really just within the last few weeks on a couple studies that I'm part of. And so in this study, I'm learning about fish and the way you grow fish. And so this study is done on lake herring, which are a little fish that you can eat. They're also fed to other fish. And it's about lake herring fry, and I learned that fry means a little fish, and it's old enough to eat by itself, so it's not just an egg anymore. And so this study was done to try to understand how these fish grow when they're fed different diets. And so this is a little mock-up, and it's relevant for the point I want to make here of the tanks where these lake herring fry, these little baby fish, live. And there's 6 compartments in each of these tanks. So you see, if you can squint there a little bit, you can see they're labeled A, B, C, D, E, F. So 6 compartments within each of the tanks where these little fish, these fry, live. And so the water's the same in each of those tanks. The water's circulating between those 6 partitions, but the fish can't circulate between the partitions. In each of those tanks, and so tank 1, tank 2, tank 3, there's 3 tanks there sort of near the forefront of that picture, the fish are fed the same diet. And so 1 diet per tank, 3 tanks. And so as I was learning about this study, so the study was actually done a few years ago, and they've brought me in to help them do some of the statistical analysis after the fact. Now, many of you maybe as practicing statisticians recognize the challenge of a situation like that. We always like as statisticians to be part of things from the beginning. But I said, hey, I've got a question for you. Why 6 sections in these tanks? And so the researcher said right away, sort of, proud, like, well, hey, replication. We know replication is important, and so we did that. And I said, well, what was the goal of the replication? I appreciate, okay, it makes sense. There's replication, 6 replicates in each tank. And the researcher said, well, replication's a good thing, right? And then I kind of paused and, well, yeah, but why did you do it? I really don't know why we did it, but we know we need to replicate. Replication must be a good thing. So then I asked another follow-up question. I said, okay, that's fine, because the gentleman felt a little bit bad that he didn't know the answer to the question. I said, well, here's another question I had. Why feed the same diet for each of the tanks? And so there's 3 tanks, and each of those 3 tanks gets a separate diet. And so you got diet number 1 in tank number 1, diet number 2 in tank number 2, diet number 3 in tank number 3, and 6 replicates right within each of those tanks. I said, well, why did you feed the same diet for each tank? He said, well, it was pretty easy because then our researchers went in and actually fed diets to the different fish. It was, you just easy tank 1 was 1 diet, et cetera, et cetera. I said, well, were you worried about confounding tank effects and diet effects? And the researcher kind of paused. Yeah, that's kind of a good point. Maybe we should have thought about this a little bit harder. I'll give you another anecdote here from a study I did, or an analysis I did here just a few weeks ago. Now, the analysis I was doing was using survival analysis. That really isn't the point to illustrate here, but I did a survival analysis. What we got was a hazard ratio of these omega-3 fatty acids. So these are fatty acids circulating in your blood, and we're trying to predict death or mortality. And you had a significant hazard ratio of 0.91. Essentially, the higher your omega-3 fatty acids, those are actually some healthy fish oils. Healthy fish generally associated with better health outcomes. And so the hazard ratio is 0.91, is statistically significant. Then I did a follow-up analysis, and this was after discussion with the researchers about this. And, well, we should adjust for some other fatty acids in the blood. Linoleic acid is an omega-6, and so based on your dietary profile, you have different mixes of these omega-3s and omega-6s. And so linoleic acids found in other things like red meat. And so we adjusted for linoleic acid in the model. And what we found is that the hazard ratio dropped a little bit, and it became more statistically significant. So the researchers saw this, and I was presenting these results. I said, look, here, model 1, model 2. The researcher said, hey, you must have made a mistake. This doesn't make sense. You adjusted for a variable, the relationship got stronger. I said, well, as it turns out, the omega-3 and omega-6 fatty acid levels are negatively associated, and omega-6 fatty acid levels seem to be independently associated with death risk, so this makes sense. And the researcher really struggled with this. And that, honestly, to this point, still doesn't quite have their head wrapped around this idea. And it's no knock on him. He's a well-experienced researcher, but this is kind of a hard idea. Omega-6s are, confounding the relationship, but really just felt like this was an impossibility. Hadn't seen anything like this happen before, where the relationship got stronger after adjusting for a potential confounding variable. I should check my code. So I hope that these two little anecdotes illustrate some common issues, and maybe you've seen these yourself in practice. Issues of replication, of confounding, of adjusting associations, and these other kinds of things that often come up in practice and are hard for experienced researchers to wrap their head around, really do have a common theme. And we would say a common theme to a lot of these questions and issues people have is multivariable thinking, both at the design stage and the analysis stage. So at this point, with that motivation in mind, we're going to transition to the next parts of our workshop this morning. And so part one, we'll turn things over to Karen, Elena, and Jamie to talk about some things going on in biology courses at Cal Poly. Then Karen is going to talk about second course in statistics, and I'll come back on a little later on in the session to talk about first courses and provide a wrap up. So with no further ado, I will turn things over to Karen.
Okay, so as Nathan said, part one of the workshop is to talk about some case studies from biology. And so the outline for this portion of the workshop is to give you some insight into some collaborations between biologists and statisticians at Cal Poly, and show you two examples of these collaborations, one, an introductory biology lab course, and then example two, some workshops that we've been running for MS students in biology. So again, these are collaborations between biology and statistics faculty, motivated by a couple of needs. We've found that students who are coming into Cal Poly often take some bio courses that use statistics before they've actually taken their required statistics course or courses. And then bio grad students who are coming into their programs often only have one statistics course, and sometimes that course was taken more than 2 or 3 years prior to entering their MS program. So they've forgotten a lot of their previous statistics. So the goals of these collaborations are to provide succinct, just-in-time materials based on sound statistics education pedagogy that bio faculty and students can leverage. And then, with these materials, to focus on multivariable thinking, things like sources of variation, design, confounding, and replication. So I'll turn it over to Jamie now to talk a little bit about the introductory biology lab course.
Thank you. So this course is the third course in a sequence of our intro series. This particular course is our physiology quarter, and they learn about plant and animal physiology. Usually the students are freshmen. Often we have some later students that are, say, changing majors, for example. Each week they have a 3-hour laboratory that discusses some aspect of plant or animal physiology, but we also have several long-term experiments that we've used this sort of multivariate idea on to try to have them think about that experiment design and their experiment data collection. This is a class that has, it's not just bio majors in there sometimes, and it's extremely broad, all of plant and animal physiology in 10 weeks. And our labs are taught by master's students and with varying statistics backgrounds. Sometimes our instructors are, quite frankly, better than me sometimes. And sometimes they, again, have not thought about it in a long time. So that can be really challenging. But this new lesson, I think, has been really effective. Next slide. Thanks. So basically what we do is we have an experiment that starts from week 1 and goes to week 5. And then we have another experiment that goes from week 5 to week 9. And on the first day of the experiment, we have worksheets that the students work through and have them think about the variables they're collecting the confounding variables and their experiment design. And then we come back together as an entire class to standardize that data collection so that all of our data can be pooled, so that we all agree on a particular design, how they measure it, what units they measure it, all of that. At the end of that 4 or 5 week period, the students use a simulation applet that will allow them to analyze those data. And the applet sort of visualizes exactly what a t-test is doing, which is really effective in turning the stats into something conceptual. Instead of a black box, you throw data into a computer and numbers come out at you. They are expected to run that t-test, graph the data, answer discussion questions about their experimental design, interpret the statistical test, etc. That initial training is done with statistics faculty so that they understand how to use that applet, and the TAs know how to do that and can communicate that well. And we do our best to align language between statistics courses and biology courses, because we don't want a situation where we're using different words to describe the same thing, and that non-alignment frustrates the students and decreases their understanding. So this has been really helpful in making sure those are aligned. We are working on doing some data collection on how well the activity helps them with their overall statistics understanding. We're doing some pre-post surveys and their attitudes towards statistics as well, and that is ongoing as we speak. So here's a sample worksheet. It's just some questions from it. Basically, the first set is, how would you set up your independent variable? Exactly where on the plant are you going to manipulate it? How are you going to manipulate it? How are you going to choose that point? And how to measure the dependent variable, same questions. How are you going to measure it from what point to what point in what units? Have them think about those sort of things. And then we also have them think about things that could influence their results. And most of the students actually do very well on that section, where we have large agreement around the class of the kinds of things that we need to consider in our experiment design, in our plant care, et cetera, on how to control those variables and how to make sure that they don't influence our results. And I think that really helps them, not just in their statistics and their multivariable thinking, but also in their overall scientific experience, that they have to think about those kinds of things when they set up an experiment. I think it's been very useful. After running it a few times, I would say that we continue to have issues with hypothesis and prediction formatting. They still get confused on that sort of thing. They also get confused on the difference between statistical significance and biological relevance. If we ask them to interpret a statistical test, they will say it's significant or it's not significant, but they won't tell us the biological relevance of that result, which is something we will continue to emphasize with them as we go forward. I think having continued collaboration between the statisticians and biologists is really important. Because we're a 2-year institution, or with our master's program, I have a lot of turnover with TAs. So we'll need to have the statisticians come back multiple times so that we can continue to have those meetings and continue to have that training. But obviously that is difficult to do. And having exact alignment between statistics and biology can be difficult because of vocabulary differences and similarities for things that are different, unfortunately. But I think that having this activity has really helped me as an instructor in terms of my statistics communication, and I think has helped our TAs talk about things outside their quote unquote wheelhouse well. So I think it's been very useful in that respect as well.
Hey, Jamie, I'm going to
Yes.
jump in. You got a question in the
Oh.
Q&A. Do you end up guiding students to a particular experimental design, or can the students make choices that lead to different experiments from quarter to quarter or between sections?
So for one experiment, we allow the one class to choose their design, because it's about plant growth and they can measure, there's many different ways to measure plant growth. For the other experiment, we guide the entire full course to one experiment design, but we have a lot of student input. So those first classes maybe get a little more leeway, but after that we go with what they've done, so that we can pool data across an entire course instead of one whole lab. All right.
I'm going to jump in on that idea of the repeated conversations between the statisticians and the biologists. So what Jamie just described is the end result of what we did. And I didn't actually track how many meetings it took us to get there, but this did occur over a number of conversations. And then, you know, we would go away and we would work on something and Jamie would develop something and we would all come back and meet again. And so it's not something that can be implemented, you know, sort of all of a sudden. It's not a once-off kind of thing. It does actually require an ongoing collaboration, but as Jamie said, we think it's really important. One of the examples of words that statisticians and biologists use differently that as a biologist, the first time I realized this, it was sort of an aha moment, was just the word model. When statisticians say model and biologists say model, they mean really totally different things. And so there's things like that that I think it's often hard to realize until you're actually having conversations with people in the other discipline. Another plug for having collaborations. Okay, are there other questions for Jamie or on the intro bio example before we move on to our next example? Okay, good. Then we'll move on to our next example, which is at sort of the other end of biology students' education. So we, as Jamie mentioned, we have a master's program in biology and it's a 2-year program. And we actually have a specific sub-program that's an interdisciplinary master's with biomedical engineering and animal science. And that's the program for which we developed the workshops that Karen and I will now talk about. So when we started this master's degree, it did not actually include any statistics requirement in the graduate program itself. We assumed that students came in with a first course in statistics, but we didn't follow up on that particularly. And the curriculum in this program is a little bit constrained already because of it being interdisciplinary and 3 different programs involved. So when we first started, we actually didn't give the students any specific statistics training. We quickly discovered that that was a problem. So we recruited Karen to start running workshops for the students. And she initially ran 3 workshops each year. So each quarter, fall, spring, and summer of the first year of the program. Since then, we decided to actually rework the curriculum so that the students are now required to take a stats course, and it would be the 2nd course in statistics. But even with them now taking that course, we are still getting Karen, at least for now, to teach 2 workshops. So we've dropped the middle one, and she'll describe what the workshops are in a second. But it was really interesting, sort of the progression of this program for us to realize how much we needed the statistics, and that even having 1 course in the curriculum was actually not enough, and that we needed the workshops as well. So I'll let Karen describe what she does in the workshops.
Okay, thanks, Elena. Okay, so as Elena said, initially when this program got off the ground, we were running 3 workshops per year. Students were coming in the fall and taking sort of a 2-hour statistics refresher. This refresher touched on multivariable thinking, the ideas of replication, and specifically the difference between technical and biological replication, and then measuring and thinking about sources of variation. We looked at graphical displays and then a review of some basic inference procedures, t-tests, the paired t-tests, confidence intervals, and that sort of thing. In the spring, then, after the students had been in the first year of their program, we had another 2-hour workshop on design of experiments. And here, in terms of multivariable thinking, we were focusing on things like inclusion criteria, direct control, random assignment, just sort of general randomization, and then the idea of confounding and scope of conclusions. And then specifically talking about blocking in experiments, and again, replication, technical versus biological replicates in particular. And then also touching a little bit on power and estimating power for your studies was also a topic of conversation in the spring. And then in the summer, we saw them again, this time for a data analysis workshop. Here, thinking about adjusting for covariates, removing block effects, analyzing block designs, and things like interaction. Here, we got into more methods, analysis of variance, regression, and logistic regression. And so all of these workshops were based on examples and had a healthy dose of Q&A interspersed as the students tried to wrap their minds back around statistics and statistical thinking. So what lessons have we learned? Some advice that we can pass on for students. For most students coming into this program anyway, it's been a year or more since their first statistics course and they've forgotten a lot. So it can be overwhelming to think about carrying out their own research study with only 1 or 2 stat classes under their belt. And so decisions about sources of variability, the design, the analysis, which all need to be made before the study is actually run, can be intimidating. Multivariable thinking is challenging, right? So again, this idea of technical versus biological replicates, other things like blocking and confounding and adjusting for covariates, all of that is pretty intense and difficult for people, you know, as they're just getting back into their master's program and learning again about statistics. For faculty, short 1 to 2 hour workshops led by statisticians may be a good strategy to increase statistical training in client departments, and not only for students, but for faculty as well. And this could lead to more collaboration between biologists and statisticians. And certainly funding makes everything easier. These workshops were funded, and so that makes it a lot easier to get somebody to come in and do these workshops 3 times a year or now 2 times a year. But I think it's worthwhile to reach out to your client departments and try to establish these connections, even if initially funding is not available. So I think we'd like to stop here and have another opportunity for some Q&A. And I guess specifically in terms of questions that we have for the audience, you know, do you all have examples of ways in which you're collaborating with other disciplines at your institutions to improve statistical understanding? And then certainly we're willing to obviously answer any questions or address any comments that you have.
Hey, I was going to follow up on one of the questions from the Q&A. It was about the intro bio course and using the worksheet questions to help students kind of discover on their own ideas about experimental design. And Beth already wrote a completely accurate answer to that, which is that certainly that is happening in the lecture portion of the course. But I would say it's also happening in the lab. The experiment where they design their own activity is not happening at the very start of the term. So they've already seen examples of experiments where they've been given a procedure, but they've been told, you know, here's how you're going to set this up. And so this isn't their very first exposure to the idea of experimental design. And I don't know if Jamie wants to jump in and elaborate on that at all.
That's accurate. So they've seen a sort of cookie cutter experiment done before in this class and previous classes. And then when they get that worksheet, they get to design their own.
And then there's another question that came in, 2 more questions came into the Q&A. So one is, students forget a lot because statistics is not embedded in the curriculum. Yes, this is true. Do other courses draw on the notions and techniques that are learned in the workshops? Yeah, so I would say this is an ongoing, the answer is yes, but we could do it much better. So I think that's true at both the undergraduate and the graduate level. So at the For the undergraduate students, Jamie actually coordinates the follow-up course, the last course in our intro series. So she could tell you a little bit about the statistics that happens in that course. But then certainly as students move on, we have a very laboratory heavy curriculum. And so in multiple lab courses, students are collecting data and then analyzing their data and interpreting their data in various ways. So we are trying to scaffold these ideas. Again, sometimes that happens more effectively than others. Jamie, do you want to talk a little about some of the later course?
Yes, so the following course in the series is Ecology Evolution, where statistics plays a lot bigger role. They're collecting their own data more often. And I have done my best to continue using the same language and providing them the same format of presenting a null hypothesis, an alternative hypothesis, and prediction together, so that that idea continues on. And we've also used the statistics applet in our statistics lab, where we talk about how to run them in the program R specifically, so that, because the program R is a black box in a lot of ways, to try and keep that understanding what's happening with these tests alive in their minds as well.
I'm going to maybe jump in as well. This is Nathan again, and just talk about some of the assessment work we've been doing. You've kind of been alluding to it, Jamie and Elena, but also just invite people. I put a link in the chat box earlier saying if you're interested in participating in some of the assessment data collection we're doing, we'll be inviting people again to participate this fall. One of the things we're trying to look at there is exactly what some of those best practices are institutionally in terms of sequencing of courses, best practices in your stats courses that provide more retention and carryover into those later courses in discipline-specific ways. We have a couple of published papers on retention and intro stats students on our ISI site. I'll post a link to that here in a minute, and it'll come up again a little later on. But I think we really need more data here to help figure out what are those things we can do in our stats courses as well as in these discipline-specific courses to provide the scaffolding to really make sure that retention improves. Because what we've seen in a paper we published a few years ago now in Surge is that students don't retain a lot after intro stats courses. That's the bottom line. And that only exacerbates this problem when students get into those discipline-specific courses. So, thanks.
So another question that came up in the Q&A is about whether we have tried using learning communities for something about this biology and statistics integration. And we have not. I think it's a really interesting idea. There's been some discussion of that. We haven't tackled the logistics of trying to set that up. Jamie mentioned in passing that there are a number of different majors who take these courses, not just biology majors, and the courses are quite large. So we're talking about a lot of students. The labs only have 24 students in them, but Jamie, how many labs do you have this quarter?
This quarter we have 12, I think.
Yeah. So the logistics of doing that is just tricky with that number of students, although I really like the idea. There's also a couple questions about what software people are using. So actually, Karen, I'm going to toss this to you. Well, I can tell you that in biology, biologists, probably like many discipline-specific areas who are not statisticians, people have their favorite software that's what they got trained on, and they don't want to learn any other software because they know how to use the one they know how to use, and statistics is not their focus. So we have kind of a split in our department. We have people who love R and people who love JUMP, and they mostly don't want to agree to all use the same one because they're very fond of the one they use. But I'm going to toss this one to Karen about the software in the workshops. Our master students introduced to both the applets. Undergrad students are using, oh, sorry, this is about the master students teaching the students. Got it. Our master students introduced to both the applets. Undergrad students will be using and software frequently used in biology.
They are introduced to the applet, yeah.
And so, Jamie, what do you use any other software in the
biology lab? In 162, the biology class that we've been talking about, we use the applet exclusively, though we do teach them how to use Excel, but not for statistical analysis. But in the following course, we use the program R, and the TAs are trained on program R.
Okay. So at Cal Poly in statistics, just generally, we also sort of are split into sort of two camps. We have faculty who like R, we have faculty who like JUMP, and then use both of these within our statistics courses. In terms of the workshops that the graduate students are coming in and seeing, I've primarily used JUMP in those workshops and have demoed JUMP and showed them how to use JUMP. But we also have a statistical consulting center on campus that the graduate students and undergraduates, frankly, can leverage. And so if they have questions about other software, and they need help with their analysis, they often try to utilize that consulting center.
Okay, several other good questions have come up, questions and comments have come up in the Q&A and in the chat. So one of them is about the COVID-19 virtual environment and how we see that affecting the workshops. So I've actually been teaching a couple of graduate seminar-like courses this quarter and have found that in the virtual environment, things like graduate workshops actually work relatively smoothly, unlike teaching undergraduates, which is dreadful as far as I'm concerned, sorry. But for the graduate workshops, I think that it still works. So I think we will continue to try to do the same things in terms of teaching the graduate students.
Okay, Nathan, should we move on here?
Yeah,
maybe?
I was just going to say that. So keep the good questions coming. We can keep the chat going, but I think we'll transition to part two. Karen, you can take us into our next discussion on multivariable thinking, looking at the second course. We'll do another Q&A at the end of that, and then I'll talk about the first course in a little while.
Thank you. Okay. So as Nathan said, yes, part two is about multivariable thinking in a second course. And so I'll talk a little bit about the second course that we're talking about here and how we're, the ideas of multivariable thinking that Nathan alluded to earlier and seeing those again in this course. We'll look at 4 examples. The first example will be just an introduction to multivariable thinking in terms of talking about sources of variability in a response. And then we'll go on and talk about reinforcing multivariable thinking throughout the course with an example on blocking, an example on adjusting for a confounding variable when that variable is categorical, and then example 4, if we have time, adjusting for a confounding variable when the variable is quantitative. So the second course in statistics that we're talking about is one that's taught here at Cal Poly to non-stat majors. So these are biology, marine science, psychology, communications, computer science students. So pretty much anybody except for a business major is going to take this particular course. It's an algebra-based course. It's required for a few majors, but the vast majority of students take it as an elective and some towards a minor in statistics. Over the last 3 to 4 years, Cal Poly's gone from teaching about 2 sections or 70 students per year to 6 sections, which equates to about 210 students per year in this course. It's often taught in a lecture lab format where there might be a couple of days of lecture and then a couple of days where students are on their computers, either in that same classroom or in a lab classroom. So the second course really is about multivariable thinking, right? And so these are the 5 multivariable thinking ideas that Nathan showed us at the beginning of the workshop. The examples that I'm going to talk about will focus on the first 4 of these. Obviously, within the second course, we talk about drawing appropriate conclusions all the time and then conveying the limitations of those conclusions. But the examples that I'll focus on here, we won't be talking about that last bullet point. Okay, so when we're teaching this course, because it is algebra based and the students are reasonably strong in math, but some, you know, still struggle with mathematical ideas and concepts. So we've tried to use data visualization as much as possible to teach the ideas of explaining variation, confounding, adjusting for another variable, as well as interaction. And then we also use simulation to reinforce the idea of the null distribution and variability arising from random assignment or random sampling. And then, of course, to reinforce that idea of statistical significance. Okay, so our first example is on histograms of birth weights in grams for all babies born in the US in January of 2016. So here's that histogram showing these weights. There's about 317,000 infants in this data set. The mean is 3,259 grams with a standard deviation of 592. So this is one of the first examples that students see when they come into the second course. Coming out of their first course, they've looked at histograms, they've looked at summary statistics, but now we ask the question, what are some reasons for the variability in birth weights? And so we'll give students an opportunity to sort of brainstorm why infants in this data set do not all have exactly the same birth weight. So some answers, students are reasonably good at this brainstorming process. They'll come up with things like genetics, whether the birth was premature, mother's nutrition, mother's stress, mother's body weight. And then we always like to throw that last bullet in there, unknown, because there could be many, many sources of variation for infant birth weights that we don't know about. So let's go ahead and include this sort of unknown category. So once we brainstorm those sources of variation, we move them into what's called a sources of variation diagram, which is essentially just a table. On the left-hand column, we're talking about the observed variation in birth weights. We've got some inclusion criteria. These are US births in the month of January. And now we're going to think about explaining some of the birth weight variation using the length of the pregnancy. All other sources of variation that we came up with then get lumped into this last column, sources of unexplained variation. So now we're breaking up these birth weights according to whether the length of the term was premature or full term. Premature is in blue in the histogram, and then full term babies are in pink or red in the histogram. And we see that the length of the term does in fact explain some of the variation that we saw in that original histogram, right? So one reason why these birth weights vary is that babies who are born premature tend to have a lower birth weight, about 2,500 grams, compared to babies that went full term with a birth weight of about 3,300 grams. But there's still variation left over inside both of those groups of babies, as evidenced by the 2 standard deviations in those groups. So we're reasonably comfortable with presenting a simple model to these students. They've come through their first statistics course. They have an understanding of means and standard deviations. And so we can begin to talk about fairly early on how we might predict birth weights. So a simple model for predicting birth weight uses the means of these 2 groups, and then we use the standard error of the model residuals as a measure of the amount of variation that's left over. And we like to point out that after accounting or explaining about 22% of the variation in birth weights using the length of term, the standard error of model residuals is smaller than that original standard deviation of 592 grams. So what does it look like when we account for a second source of variability in the model? So we can move one of those sources that was in the unexplained variation column over into the sources of explained variation, and so that's what we've done with the mother's BMI. In the lower left-hand column, or sorry, of your screen, you'll see a graph of the birth weights against mom's BMI, color-coded according to the length of the pregnancy. And we see a slight increase in birth weight with an increase in mom's BMI. And now the leftover or residual variation has come down even a little bit more from the previous model. This model explains about 23.5% of the variation. So we haven't done any inference yet. We're just getting students used to thinking about explaining variation. We've talked about some sums of squares. We've talked about R squared, and then residuals in the course up to this point. So our second example now is looking at a designed experiment. We're investigating 3 stimulants: caffeine, theobromine, which is a chemical in chocolate, and a placebo. The response is the finger tapping rate of individuals measured 2 hours after they've ingested the stimulant. So again, we ask this question: what are some potential sources of variation in finger tapping rates? Give students an opportunity to brainstorm some of these potential sources of variation, and they'll come up with things like this, right? So often the first one is sort of hard to get them to say, but the type of pill or the stimulant, we are actually hopeful that that one's going to explain variation in the response. And then things like previous caffeine use that day, the size or the amount of the pill, the time after ingestion at which the test is run, the time of day, the manufacturer, age of the participants, their inherent finger tapping ability, their attitude or willingness to participate, how much sleep they got, metabolism, and again, unknown sources. So here's an analysis of a 1-variable model of finger tapping rates versus stimulants. And in the summary statistics, we see that the mean for theobromine is about 481 taps per minute. The mean for caffeine is 479 taps per minute, and the mean for placebo is 462 taps per minute. Looking at the F statistic, we have a small F statistic of 0.68 with a large p-value of 0.533. So we asked the students, why do you think that F statistic is so small? So we can point to the fact that the means do look a little bit different here, at least in terms of theobromine versus placebo and caffeine versus placebo. So we have about 20 tap per minute difference there. And so why is this F statistic so small? Students hopefully by this point in the course can look at those box plots and see that there's lots of overlap in the box plots. And so that's an indication that we have lots of unexplained variation coming from all those sources that we brainstormed a minute ago. If we put those sources of variation into our sources of variation diagram, we see that we're using the type of pill as our explained source of variation, but we have lots of unexplained sources. We have accounted for age through inclusion criteria in this study, and through the design protocol, we've accounted for training the participants on the way to do the finger tapping, and we've controlled when the measurements of finger tapping were taken 2 hours after taking the pill, but that still leaves lots of unexplained variation. And a large portion of those sources come from the person that's participating in the study. So how can we account for that person-to-person variation? In this actual study, the 4 participants served as blocks. Each participant was measured using each stimulant in random order. So this was a block or repeated measures study design where we blocked on person. So what we're going to do then is we're going to take all of that person-to-person variation and we're going to move it into a source of explained variation, and that in turn is going to reduce the sources of unexplained variation, perhaps giving us now a statistically significant F statistic. So there's two ways that we can move that person-to-person variation out of that unexplained column. We can do that through the simulation. We need to adjust the simulation to reflect the within-person random assignment that was used in the study. We can also use a more mathematical technique, accounting for the person effects in the finger tapping rates. So I'm going to switch over to an applet, hopefully here, there it is. So this is one of the applets in the ROS. Oh, you're not seeing it. Why not? No.
Did you share application or general?
There we go. Can you see that?
Yes, thank you.
Okay, thank you. So this is one of the applets in the Rossman-Chance applet collection, the one blocking variable applet, and I've got the data pasted into this applet. This first graph that you're looking at has the 3 different treatment groups, theobromine, caffeine, and placebo, and the observed finger tapping rates here are identified according to the participants A, B, C, and D. So you can see that these are also color-coded according to participant. In the show shuffle options side of the applet, we have a choice here to either use the completely randomized shuffling procedure or a within blocks shuffling procedure. And we hope that the students can tell us that if we want to account for that person-to-person variation, that we need to repeat the random assignment that was used in the actual study, which means we need to shuffle within blocks. So here's what that shuffling looks like on a participant by participant basis. We can see that each of the finger tapping rates for each participant are shuffled up and then reassigned back to one of the 3 treatment groups, theobromine, caffeine, or placebo. The F statistic from each of these re-random assignments is being plotted in the graph on the right-hand side of your screen. And if you notice, these F statistics are very small. Because the shuffling is being done within each participant, it would be impossible for all of the largest finger tapping rates to end up in one treatment group, and impossible for all of the smallest finger tapping rates to end up in the other treatment group. So it keeps the means of these treatment groups much closer together and smaller F statistics. So if we repeat this process some large number of times, I'll set it to 1,000 total, we can get a more appropriate null distribution to assess the statistical significance of the observed F statistic, which was 0.68. So this null distribution now has used restricted random assignment. And so that original F statistic of 0.68, which did not take into account the person-to-person variation, can be judged against this null distribution, and that results in a smaller p-value than what we were seeing a few minutes ago. So going back then to the PowerPoint presentation, right? The idea then, in terms of shuffling within blocks, is that we want to take those original data values and shuffle within each person. So participant A has the values 472, 453, and 446.
Karen, you're still projecting the applet.
Sorry, I haven't quite figured out how to get back and forth here. Thank you. Okay, so we will shuffle within each person, and then after each of those shuffles, compute the F statistic and then create a null distribution based on restricted randomization against which we can judge the significance of that F statistic of 0.68. So the second way within a block design to account for that person-to-person variation is by removing the person effects. So I'm going to go out on a limb here and see if I can get you back to the applet. So back in the applet now, we have participant D again is in blue, and we can see that participant D has the fastest tapping rates, the most taps per minute, while participant A has almost the fewest taps per minute regardless of the treatment groups. I'll show you the block effects here. The block effect for participant D is 36 taps per minute. This means that on average, participant D is tapping at about 36 taps per minute more than the overall average, whereas the effect of participant A is -17, meaning this participant is tapping at about 17 taps per minute slower than the overall average. And then we can talk about adjusting these data values for the person effects. So that literally means that for each observed tapping rate for participant D, we're going to subtract 36 taps per minute. And for each observed tapping rate for participant A, we're going to subtract -17 taps per minute. When we do that, when we adjust the data for the block effects, we see that the adjusted data values converge towards the means of each of the treatment groups, and there's much less within group variation now after removing the effects of each individual. So to recap, we can account for the person-to-person variation by removing those block effects. Because the randomized complete block design is a balanced design, the treatment means stay the same, but that within treatment variation decreases substantially. Here are the side-by-side ANOVA tables. The one on the left is that original analysis of variance. The one on the right includes the block row where we've adjusted for a person. Looking at the pie charts at the bottom, we see on the left that before adjusting for the block effects, about 87% of the variation was unexplained. After adjusting for block effects on the right, we see that only about 5% of the variation now is unexplained. This has had the result then of increasing that F statistic for our stimulant treatment and giving us a small statistically significant p-value. Okay, so still, on this idea of multivariable thinking, but switching now to an observational study and confounding. So example 3 is going to use car accelerations. Acceleration is the time in seconds it takes a car to accelerate to 60 miles per hour. So here's a histogram of those accelerations. That histogram is centered at about 15 seconds with a standard deviation of 2.76 seconds. And so we might ask again, what features of a car will explain faster acceleration times? Here's a potential sources of variation diagram. The observed variation is accelerations. We can think about maybe the weight of the car might explain some variation in acceleration, and it does, about 6.7%. There's other sources of unexplained variation, horsepower, number of cylinders, hybrid or not, although maybe not for 1983 car makes and models, but certainly that would be true today. Let's look at a few pairwise relationships now. So the first graph shows acceleration versus the weight of the car. And we see sort of an unintuitive relationship here. So it turns out in this data set that heavy cars actually have faster acceleration times on average than light cars. So the mean acceleration for heavy cars is about 14.8 seconds, and the mean acceleration for light cars is 16.2 seconds. The analysis of variance shows that we have a statistically significant association between acceleration and weight of the car. And so we've established this sort of initial relationship. But what about the horsepower of the car? Looking at the way acceleration relates to horsepower, we see that high horsepower cars tend to accelerate faster than low horsepower cars. So this is the relationship that we would expect to see. And so now we've established a relationship between acceleration and horsepower. How does horsepower relate to car weight? These are both categorical variables, binary categorical variables. And so looking at a mosaic plot, we see that the light cars tend to be low horsepower cars in this data set, and the heavy cars tend to be high horsepower cars. So we've now established an association between horsepower and car weight, and this then establishes horsepower as a confounding variable in the relationship between acceleration and car weight. So what does it look like when we add horsepower to the model? Well, we have to keep in mind that we have this pretty strong association between car weight and horsepower. And so when we add horsepower into the model, we want students to be able to tell us that this original relationship between acceleration and car weight is probably going to change in some way. So what does it look like when we adjust for horsepower in this model? So here's the original association between acceleration and weight. You have light cars on the left and heavy cars on the right, and this unintuitive, in terms of direction, relationship between acceleration and weight, where light cars are taking longer to accelerate on average than heavy cars. Color coding now according to the horsepower of the cars. So the red dots are low horsepower cars. The blue dots are high horsepower cars. And so we will ask students, if we remove the effect of horsepower from acceleration, what will happen to the blue dots and what will happen to the red dots? So this is analogous to removing the effect of person from the tapping rates in the previous example. And so we want to remind students that if we're talking about the blue dots, those are high horsepower cars, and high horsepower cars tend to have faster or lower times. So if we remove the effect of horsepower from those cars, right, those acceleration times should shift up. What about the red dots? The red dots are low horsepower cars, and low horsepower cars have slower times. So if we remove the effect of horsepower from acceleration, those red dots should shift down. So now looking at the lightweight cars in particular, we have to remember that there are more low power cars, a higher proportion of low power cars than high horsepower cars in this data set. So we're going to see more of those red dots moving down than blue dots moving up. And so the net adjustment on that mean of 16.25 should be a shift in the downward direction. Looking at the heavyweight cars, we see a much larger proportion of these cars are high horsepower cars. A lower proportion are low horsepower cars. So we're going to have more of those blue dots that are moving up than red dots that are moving down. And so the net change on that mean of 14.83 should be up. So I'm showing you screen captures from another Rossmann-Chance applet, and we have the ability then to show this shifting up and down of these points, and then the resulting adjusted means of 14.69 and 15.84. And so now, right, the direction of the association between the horsepower adjusted acceleration and weight is in what we might consider a more appropriate direction, with lightweight cars accelerating faster than heavyweight cars. The association, though, is a little bit less strong than it was before adjusting for horsepower. So the F statistic for weight is now about 12.52. I think previously it was around 28. So we have a slightly less strong association, but an association that's in the correct direction after adjusting for this confounding variable horsepower. So the last example is again looking at car acceleration, this time in the context of regression. And so all of these variables are continuous in nature. We have acceleration versus the weight of the car, and we have this unintuitive negative association between those two variables. The second plot is horsepower versus weight. We have sort of a moderately strong relationship between horsepower and weight in the positive direction. And then acceleration versus horsepower, again, sort of moderately strong, but in the negative direction. So again, we have this association between acceleration and car weight, and then we have the individual associations of horsepower with acceleration and horsepower with car weight, establishing horsepower as a confounding variable. Now, adjusting again for horsepower. Horsepower is a continuous or quantitative variable, so we've simply color-coded horsepower with higher horsepower cars in sort of the blue coloring and low horsepower cars in the darker coloring. And if you look at cars that have the same horsepower, we see sort of these positive associations between acceleration and weight. So this is an example of Simpson's paradox. And when we adjust those accelerations for horsepower, we see sort of this up and down shifting. So a shift up for high horsepower cars and a shift down in acceleration for low horsepower cars. Because weight is also quantitative, we need to adjust the weights for horsepower. This is going to be either a left or right shift according to horsepower. So high horsepower cars tend to be heavier, so when we adjust for horsepower, those cars should shift to the left. The low horsepower cars tend to be a little bit lighter, and so when we adjust for horsepower, those cars should shift to the right in terms of their weights. So now if we put that all together and adjust for both, in both directions, we adjust the accelerations for horsepower and the weights for horsepower, and then fit a new regression line, we see a change in the direction of that relationship between the horsepower adjusted accelerations and the horsepower adjusted weights. So we've looked at 4 examples, and in summary, in the second course, we've made thinking about sources of variation an important skill in both randomized experiments and observational studies. And we try to sort of go back and forth between these 2 types of studies within the course. We leverage simple data visualizations to show what it means to adjust for a variable. And these data visualizations are in applets that students can play with and watch this adjustment process happening as they try to learn and predict what things will change, what things won't change as we adjust for either a blocking variable or a confounding variable. And then we use simulation to reinforce the idea of the null distribution and that variation that arises from random sampling or random assignment, and then how that relates to statistical significance, with an emphasis on the simulation matching the design. And of course, there's additional multivariable topics that are covered in the second course, particularly interaction, both in factorial studies and in regression models, and also collinearity. So I think I'll stop here and open it up for questions and comments. And a couple of questions that we have, right, are listed for you here. The second course is all about multivariable thinking, but it's often taught as disparate analysis methods. So for example, we have a unit on t-tests, or we have a unit on ANOVA, or a unit on regression. So what are some ways that you all try to weave this all together to create a unified approach? And then also, are there other important multivariable ideas that need to be stressed in the second course so that students who only see 2 courses in statistics can be successful in a data-centric world and job market? Okay, so I'll ask maybe Nathan to come back in, and perhaps he can help field questions and answer questions.
Sure, absolutely. So yeah, continue to ask those questions through the Q&A option or through the chat window. I know John Melendez is asking for data on COVID-19 which could be pulled into your courses. I know there was a recent conversation about this, I think on the ISOStat listserv, but I'm forgetting what some of those links were. So if others want to post those, feel free. There was a good discussion there also about how to do that well in the context of your class in terms of, not burdening students with it, but also, yeah, kind of walking that line of including things that are relevant and timely, but also not, making it uncomfortable for students.
Karen, do you want to say any more about kind of the questions we were answering earlier about applets versus technology and how you weave those together in the second course?
Sure. So we do want students to be able to do some statistics and do some statistical analysis when they leave the course. I like the applets that we've developed in terms of, demonstrating to students how this adjustment process is working. So that they can actually see beyond just changes in summary statistics and changes in F statistics and confidence intervals and the like, how we get to this sort of shifting up and down or left and right when we adjust for a confounder, or in a block design, how that variation within the treatment groups becomes so much smaller when we remove the effects of blocks. So I think the applets are really great in sort of, getting these really complicated ideas across to students in a very simple and intuitive way. So I often will start with the applets and let them play with the applets to learn these sort of concepts, and then gradually move away from the applets as they get comfortable with the ideas and the concepts, and show them how to do these analyses in JUMP. And I know that Nathan and some other folks on the team also, do something similar, I think, but use R rather than JUMP.
Yeah, I'll just comment real briefly. And Karen, there's a couple other good questions in the Q&A window I'll let you maybe jump to. But briefly, yeah, I mean, so I'm using R along with the applets. And I, just to kind of amplify what Karen said, I think when I first started teaching the second course, thought it would be primarily about what I would call end user software. So R, JUMP, whatever it is, and really have shifted my thinking to focus and say, I really want my students to learn important aspects of multivariable thinking in that course, and that intuitive applets, the ones that Karen was demonstrating, is the best way to do that. And I certainly ramp up the amount of R that they're going to do versus the first course, but it's still less prominent than I might have thought it was a few years ago when I started teaching that course and really thinking hard about it.
Good. So there's a question about what would be the downsides of doing all ANOVA-like situations via regression with indicator variables, so not using ANOVA at all, or to at least introduce it all via regression and then later say how that's often phrased in terms of ANOVA. I think that's certainly a valid way to do things, and we kicked this around a lot within our team when we were sort of developing our curriculum. I think one important thing that we try to do within this course is as much as possible to not separate ANOVA from regression. We use ANOVA as a way to talk about explaining variation and the sums of squares that are provided in an ANOVA table are used to compute, sort of R-squared statistics that we can talk about the amount or proportion of variation that's explained by each variable. And that's regardless of whether that variable is a quantitative variable or a categorical variable, and regardless of whether the model that was fit was sort of this separate means or effects model from ANOVA versus a regression model from, a regression analysis. So we try to make, this idea that each part of the analysis is contributing very important information. You have sort of the model part of the analysis, and you have the ANOVA part of the analysis, and those are complements of the same analysis.
And maybe just jumping in on that one too, Karen. I think I probably too initially would have thought, I'm going to focus most on regression. And I think when we, Karen, you showed the sources of variation diagram, and I love that way to help students start thinking about a study in a deep way. I'm going to talk about that in a minute, even in the first course, about how you can do that. And then the parallels there that segue right into ANOVA and explaining those sources of variation. I just really like that. And I think that for me to think, I guess my thinking has developed to the point where I say ANOVA's got these benefits to thinking about a study, and a regression model has these benefits to thinking about a study, and why not try to weave those as much as we can simultaneously back and forth in front of students, so that students leave the course realizing two different ways of looking at the same problem, but it's, they're sort of parallel methods. And I think that there's a lot of benefits to that approach.
So I know there's been a couple of questions or comments about how can this be done in JUMP, and certainly Graph Builder in JUMP is a great tool because it's just drag and drop to create these different graphs. I think in terms of seeing the adjustment process, that's where the applets really come into play, and would strongly advocate stepping away from a software package and into the applets to demo that sort of adjustment process. And then I think one final question here, that there was a comment that there was only 4 subjects in one of the studies that we demoed for you. And that's true. And we do talk about sample size and the importance of sample size. But it's also true that lots of experiments, particularly block designs, do use pretty small sample sizes. And so I think it's important to show students that we are making decisions about important things from small sample sizes, as well as decisions from studies that use larger sample sizes. So I think we try to show a really wide variety of studies throughout the course to give students insight into all these different issues that come up.
Yeah, I mean, I think just to that, sometimes a small sample size is good enough and sometimes it's not. And if my students can leave appreciating those distinctions, that's great. I know that some additional questions are coming in. I think in the interest of time, I'm going to segue and get to our last part here, and then we'll have more Q&A time here at the end in a few minutes. And so, as we sort of walked you through over the last little while, we started talking about some of the problems we're seeing in practice. We then talked about what might you do with students in their discipline, in this case, biology. And then Karen just talked about the second course, and I'm kind of walking back now to the very first course in statistics, which obviously for some of our students might be the only course they take in statistics before they head off into their discipline. Maybe we can get more students going into a second course like Karen was talking about. But I'll just share with you briefly a few things we're doing to start anticipating big ideas like multivariable thinking in a first course in statistics without getting students, what I'll say, is stuck in the deep end too fast. And so a kind of a summary of my thoughts, and then I'll unpack a few of these, and then again, we'll have some more time for Q&A here in a few minutes. Not spending a lot of time on data wrangling and cleaning missing values and R coding and things like that. Those things have a place. We could debate how much of a place they have in the first course, but I think as much as possible, making sure that they don't have a large place. I think then what should take a large place, I think it's focusing on the big picture of statistical investigations. I'll walk you through an example here in a minute of what I mean by that. Focusing on the core logic of statistical inference, anticipating those themes of explained variation that Karen was talking about and you heard earlier in the session as well. Getting students experience on real data analysis projects, I think is wonderful, and really trying to excite students for a second course and/or courses in their disciplines, leaving them with a sense of empowerment and excitement, that thing we've maybe heard before, knowing when to call a statistician, even if you're not going to call yourself one, and we don't expect them to after a first course. So those are kind of the big things that I like to do in my first course. So what can this look like? Well, the very first thing that I do with my students on day one, right when they get there, is we walk through the 6 steps of a statistical investigation. I've got those up on the screen for you. So asking a research question, designing a study, exploring the data, drawing inferences, formulating conclusions, and looking back and ahead, and telling students that throughout the course, and then really operationalizing this as well, throughout the course, every study we look at, we're going to talk about these 6 steps. This is the big picture of statistical investigations. And as I walk through this one with you, I hope that you can appreciate how this, I think, helps students focus on statistical thinking and multivariable thinking. So this particular study comes from Science. You see here real data in a real published journal, accessible for students though. The background is that 85% of Americans approve of organ donation in principle, but only 38% of Americans are registered organ donors. And so the question this article from Science was proposing is, can a method be found to increase the likelihood that a person agrees to become an organ donor? So we talk about this with students, and then I walk them through the study design and say, so the researchers recruited 161 people online, asked them to pretend to apply for a driver's license. They weren't told the real motivation for the study. They just said, all you have to do is fill out this questionnaire. As it turns out, the researchers randomly assigned people to one of 3 groups, the opt-in, opt-out, or neutral, or forced choice groups. This is the way you asked people to become an organ donor. It was either check here to be an organ donor, check here to not be an organ donor, or do you want to be an organ donor? You must answer either yes or no. And so this graph we can show our students even on day 1 of a course, just looking at the percents and say, hey, the people in the opt-out or the neutral group had higher rates of becoming organ donors than those who are in the opt-in groups. The opt-in group would be that check here if you want to become an organ donor group. And they were the lowest percentage. Now, later in the course, we get into step 4. On day 1, I might just sort of anticipate this idea of how do you know that those differences of 70, 80% versus 40% is big enough to be meaningful statistically and not just something that would happen by chance. And so that's something we're going to unpack later in the course. Later in the course, we're going to introduce students to what we call the 3S process. And even with a more complex study design like this, with 2 variables and 3 groups, we try to show students using simulation. Karen talked about this. I'm going to go very quickly here, but just how you can use, in this case, the mean absolute difference in those pairwise proportions. You can shuffle using some of the same applets we've been talking about in the Rossman-Chance collection to evaluate this data and see that we do get a statistically significant result. Harkening back to what we were talking about earlier when Jamie was talking about the contrast with the biology students of statistical significance and practical importance, this is something you can also start talking about on day 1 with your students. And then we also would show students as part of step 4 some confidence intervals. And then step 5 is generalizing, and so asking who are these 161 people, and can we say cause and effect, and why can we do that here? And even on day 1 of your course to start anticipating these kinds of questions. And so I'll open it up to students after a brief introduction to the study. What do you think about this study? How might this impact your thinking? What questions do you have about the design? What questions do you have about even just that descriptive analysis with the bar chart? What might next steps be? These are all important questions to get in front of our students early and then emphasize throughout the course. I do think that this 6-step framework allows you to continually hold these ideas up in front of students. And really step 6, and to me this is in a lot of ways the most important place to end. We so often forget to do this when we're stuck on a particular day in our class and we're running short of time. We might sacrifice the last step, right? We might spend more time, my students are going to get confused about this way of thinking about sampling distributions. I'm going to spend a lot of time there. And that's certainly important, but making sure we always land on step 6, looking back, what did you do? and how did you do it in your study design? And then looking ahead, what would you do different next time? What are the implications of the study for practice? What are the ethics of some of these? Students have great conversations on day one about the ethics of requiring people to, answer those questions, or, having the default be an opt-out where you're going to be an organ donor if you don't check the box, and confusing question wording and this kind of stuff. And then the fact that this was a hypothetical scenario. Hypothetically, fill out this driver's license application. How might this look different in practice? And even in the first day, laying some of the foundations for random assignment and the impact on confounding variables. One of the things that I'm playing around with more now is even thinking about using that sources of variation diagram that Karen showed you and using it in a first course with my intro stat students. And really, I don't think there's any reason why we couldn't do this. I'm trying to figure out, how do I weave that in well to lay a foundation of explain variation and things like inclusion criteria early. I really think in the context of my first course, this kind of brainstorming about sources of variation is going to help them answer those step 6 questions. So what would you do different next time? What are the implications of this? The limitations of your study design, or the benefits of your study design. In this case, it's a randomized experiment. What kind of benefits does that have in terms of explaining away some of those sources of variation by dealing with confounding? So to sort of conclude this little dive into the organ donation example, what I'm going to do on day 1 with this example is going to be different than what I do later in the course when we come back to it. On day 1, I'm going to emphasize the study design's importance, and I'm going to really spend a lot of time on day 1 talking about step 6. One of my primary goals in the first day, first week, first 2 weeks of the course is to get students asking questions about statistics and feeling like they have something to contribute to the classroom. So by asking students to critique the study design, thinking about the ethical and moral implications of the ways that we're going to recruit people to be organ donors, thinking about the limitations of the study and who these 161 participants were, those questions are what I'm going to call level the playing field questions. So students come into my class from a variety of backgrounds. Many of them are pretty math phobic. Many of them don't have strong quantitative backgrounds. So by asking these kinds of questions early, those students are able to contribute. We don't end up with as many, 2 or 3 students at the top of the class who are really quantitatively mature, answering all the questions, setting up this dichotomy from day 1. We're going to level the playing field by asking these kinds of questions early, and then continuing that trajectory throughout the course as we spiral over and dig into some of those steps in more detail. So I hope that that keeps students out of those bogs where we end up with the students who think everything's black and white and a right answer or a wrong answer. And this is really complex. I don't know, it's like a magic black box, and I'm not going to trust most of it because it seems like it's too magical to me. So threading the needle here on complexity. And then later in the course, getting back to more these ideas of generalizability, confounding, the implications of the randomization here, and et cetera. So what does multivariable thinking look like in a first course? That it recognizes that variation in response comes from many sources. Again, that's that step 6. I think it's also thinking about generalizability and those other variables that are going to be characteristics of your observational units, and how do those generalize to a broader population or not. It's thinking about causation and the handling of confounding variables or not, and what implications that might have on your study design. I also think it comes from exploring data, and we're starting to do more with multivariable graphs with our first course in statistics students. Karen showed you some examples, I'm not going to here, but coloring that third variable on your scatter plot, or adding a third variable into your contingency table and things like that. Talking about Simpson's paradox, and that's been there for a while, maybe making that a little bit more prominent. These are all ways of starting to do some multivariable thinking with your students in a first course to lay this foundation from which they can then go into their later courses in their discipline or in your statistics program. Other quick tips, and then we'll segue to Q&A. So if you got those questions, you can start getting those in the boxes there through the chat or through the Q&A window during our last few minutes here. Real data analysis project, I think, can be really helpful. And also just keeping in mind that I think for me anyway, one of my main goals is trying to excite students for further learning. Realizing that this is real data that matters. It's not all cutesy M&Ms. That organ donation study is a real study done. It was published in Science. It has real policy implications for the way our world works. Keeping it focused on the big picture, not lost in the weeds, and trying to thread the needle on this black and white view of statistics. It's either right or it's wrong, and versus statistics can say anything. So in conclusion, I do just want to acknowledge we've been fortunate to have some National Science Foundation funding for the STUB project for some of the curriculum development for both the first course and the second course. A wonderful team. You've heard from some of them here today, a number of others listed on the screen there. To learn more, isi-stats.com. You can link to some of our curriculum materials. Some of those are for free. You can also link out to see ways to sort of get copies of some of the published curricula that are there. All the applets are free. Beth posted that link, rossmanchance.com, or you can go through isistats.com. We've got the second edition of the first course book, first edition of the second course are coming soon. 2 websites on CAUSE-Web that are worth potentially looking at: the STUB website for statistical thinking and biology, the SBI site to learn more about simulation-based inference. And we do have a few different assessment initiatives going on right now for the second course, the first course, as well as those biology courses. Feel free to email myself or Beth for more information on those. And so with that, I will wrap up and say we've got a few minutes for some concluding questions, either on the first course or going back to any of those earlier parts, the second course or the biology discussion we had earlier on, or anything else about multivariable thinking. We'd love to hear from you here in our last few minutes together. So one of the questions from Julie just came in: returning to the theme of real data, but important and relevant questions, what suggestions do you have for identifying good data sets? That's a great question. I would say that when I started trying to get real data that matters, I was really a little bit, it was a lot harder than I thought, because so many of those real data studies that are published are really complicated and way beyond typical students. What I often am doing now is, as I'm reading the news and looking at the things that are, or listening to the news, the things that are published there in the research sessions, they've often taken studies that they think are going to have broad impact across society. It's enough for sort of a news bite as a, in the health section or the research section of the news organization. And then going and finding those studies and the actual peer-reviewed journal article. And then some of those can be pretty good for students. And I'm sort of leveraging the fact that the news organization has already done a lot of the filtering and distilling down. I think also just being on some listservs. ISOSTAT's a wonderful listserv for great timely ideas. And if you just go on there, or some of the other listservs the ASA has, the stat ed section, also the Journal of Statistics Education Data and Stories section. I'm sure others have other ideas as well about where to get good data sets.
Nathan, we have one other question from about multivariable thinking, and someone asks, wouldn't a huge impact on emphasizing multivariable thinking be made if it was part of the AP statistics curriculum? And they note that except for the occasional free response question, that multivariable thinking isn't part of that curriculum.
Yeah, I think you're right, Paul. And when I think about the first course in statistics, the standard curriculum for that is somewhat driven by AP, but I think if we continue to innovate in the first course and do more with multivariable thinking there, and the GAISE Guidelines is setting a standard, this is part of what needs to happen, then I think as we innovate more there, hopefully that works its way back to AP. But I would also encourage us to realize that things like confounding, and the impact of a third variable and Simpson's paradox, and some of those things have been there for a while, and so it might just be emphasizing those things a little bit more. I think one thing that hasn't been there is an emphasis on study design quite as much, especially randomized experiments and what they get you in terms of confounding, and that's probably something we could emphasize a little bit more.
Great. Well, unfortunately, we've run out of time, but I want to thank all the panelists for really a great workshop today.